All right. So yes, my name is Anthony Burnetti. I work with Will Ratcliffe here at Georgia Tech. Ordinarily, I work with yeast most of the time, but and we actually study the evolution of multicellularity using yeast. But that got me interested in studying other major evolutionary transitions, not necessarily in the lab, but by looking at the history of life as it exists on Earth. And I think there's some very interesting things that you can tell. So the big question that got interesting to me was, are major evolutionary innovations and transitions predictable, or are they contingent? And people have been trying to answer this question forever, and everyone's looking at the same thing and coming to wildly different conclusions. There's this wonderful book from Monod in the 70s where he looks at bacterial biochemistry and says, oh my God, this is so bizarre. We finally know we're alone in the universe. And other people look at the whole history of life on Earth and say, okay, this makes sense. Why would this not be something that would just straightforwardly happen? And so what this tells me is that people are not looking at this systematically. They are using wildly varying intuitions. And so I think it would be useful to try to look at this systematically and figure out what's actually going on. So there are some major evolutionary innovations that appear again and again and again. And this is a beautiful tree of the eukaryotes. Every clade in there that has any yellow on it has multicellular instances, and that's not derived from the beginning. That's every one of those has separately invented it. It just happens over and over, and so this is fairly predictable. This is something that is easy. It happens all the time. But there's a lot of innovations that people call, well, not a lot, there are multiple innovations that people call evolutionary singularities. These are things that happened once and only once and changed the world, and people are very interested in these. The classic one is, of course, origin of the translation of genetic code. That's a heck of a big deal. Origin of eukaryotes is very important. And so why are these things singular? These could be extremely unlikely to evolve, and we only see them because of an anthropic selection effect, like we're here because interesting things happened, meaning that we can look at it. Or there could be some kind of a historical process that limits what you can actually see and what is there today. So how can we look at this? And I'm going to look at this by looking at phototrophy. Phototrophy is the ability to use light for biological energy. And I would argue that this is the evolutionary innovation after translation itself, basically, because non-phototrophic primary production is barely any of the total biomass of Earth. The vast majority of biomass comes from using light to fix carbon. And it's not a coincidence that the earliest evidence of life on Earth that nobody disagrees with is photosynthetic microbial mats. This is not an accident. It changed everything. It takes the Earth from something where you might not notice it was alive if you were standing on it to something where you can see that the atmosphere is weird across the galaxy with a good instrument. So I would say this is fairly important. The interesting thing, though, phototrophy was invented twice. Not once, it's not singular, but it's not like multicellularity with it being invented a bunch of times. And two is interesting. It provides information because you can compare the two against each other, but it's still rare. It means that there is something limiting the number of instances that you see. And so I think this is an important window into things that are truly singular. And this has happened, like I said, twice. It's happened in retinalophototrophs and chlorophototrophs. Chlorophototrophs, everything green you see outside is a subset of that. And retinalophototrophs, they're generally purple. They're all microbes. You see them flying over salt flats is where you're most likely to actually see them in your day-to-day life. So, like I said, very important window into evolutionary innovation. I want to see why we're looking at two, why we're seeing two, and also how they compare to each other. So this is the wrong file. So let's see. This is I am pretty sure that this is indeed the wrong file. Can I put something on from my flash drive real quick? Okay, I got it. I got it from here. So let's see. Or is that the other one? Got two flash drives on my keychain. There we are. Got it. There we go. Sorry about the slight issue there. So let's take a look at the two different instances of phototrophy on Earth. So the first one I'm going to look at is microbial rhodopsins. This is what mediates phototrophy. This system is just a single 27 kilodalton membrane protein. It's got a little dye molecule in it. It absorbs one photon and pumps one proton every time that it runs. And it gathers a small fraction of the energy of that photon, and it's really simple. This is the entire functional unit. You need 2 to 5 genes to make the pigment. So this horizontally transfers like mad. It's all over the place. It's in bacteria, archaea, eukaryotes, everywhere. And this is a nice way to just get a little bit of energy from light. Then you've got the chlorophotrophic reaction centers. These are extraordinarily different. These are horribly complicated. This is oxygenic photosynthesis and many other groups of bacteria. You've got dozens of chlorophyll and bacterial chlorophyll pigments, other things, redox cofactors, everything in an enormous multi-protein complex, which is itself part of an electron transport chain. So, and this moves around electrons via photochemistry. And so by plugging this into an electron transport chain, every photon you absorb, you can pump 2 to 4 protons across the membrane and charge it up. And it depends on the exact electron transport chain you're plugged into. It's super flexible. And you can also use it to fix carbon, because you can shoot electrons up to energy levels that can be used to reduce the CO2. And in order to horizontally transfer this, you need to transfer 30 genes. It does happen over evolutionary time, but it's rare that you can look at a tree and of life and count the number of times it probably happened. So these things are very different. And yes, this is the most complicated system. This is the one that actually makes oxygen. It gets as simple as that. So no, this is much more complicated than the other version. So let's sort of abstract them down a little bit. They're just very different systems. You've got this simple system that just pumps 1 proton per photon. You've got this enormous system that also plugs into antenna pigments that sort of funnel light into it and plugs into an electron transport chain. And these things are different in so many ways, in the light they absorb, the elements that are in them. I'm not going to talk about that right now. I'm just going to talk about one particular difference between them. And that has to do with their energy flux and yield versus their mass. And you get this just by looking at the size of the system, how fast it turns over, and what it produces, and bringing these numbers together. And it turns out this tells you something super interesting. This is a graph of the energy flux per unit mass when the system is running full tilt. I dug up a bunch of information on a bunch of different systems. I got 2 chlorophotrophic systems and 2 retinalophotrophic systems. The retinal's churning energy like crazy per unit mass when it's at saturation. And that's because, yeah, they're not as good, they're inefficient, they run slower, but they're tiny. You get an enormous flux per unit mass. It's really low fluxes per unit mass for the chlorophyll. And so you get an enormous return on investment for the same mass of rhodopsin versus a chlorophototrophic system. But at the same time, this larger machinery, it also absorbs a lot more light. It's full of lots and lots of pigment. And so if you actually look at them at different light levels, at low light, the chlorophyll wins hands down. These two lines, if at really low levels, this orange line here actually wins handily. At low light levels, the chlorophyll wins handily. At high light levels, the retinal wins handily. So the chlorophototrophes are efficient per incident photon, but then they saturate. The retinal is efficient per unit infrastructure when the resource is abundant. So now this is looking at a couple of systems that exist in life today. I can do better than this. I have actually managed to create this simplified numerical model of just a generic phototrophic system. This can be applied to either chlorophototrophes or retinalophototrophs. You take a central engine as a given in orange here. It's got a certain amount of absorption it can do. It has a velocity, a yield, and then you have different amounts of antennas. And this is a feature of everything that uses chlorophyll and some things that use retinal. And then this whole machinery gets degraded. I've parameterized all of this based on work of 15 other groups who have done amazing experiments. And what I'm able to do is I'm able to vary the light level and allow the amount of antenna to vary and create a numerical solution for the optimal chlorophototroph and the optimal retinalophototroph for a given light intensity, which is sort of an ecological sub-niche. When I do that, this is what I get. This is light intensity from super low all the way up to direct sunlight at the equator. And this is the efficiency per unit light of the two systems and efficiency per unit protein of the two systems. So at low light, once again, the chlorophyll wins hands down. At high light, the retinal wins hands down. And when I actually look at their compositions, this is the amount of antenna associated with each system. At their crossover point, this amount of antenna is within 30% of the smallest known antenna of actual chlorophototrophic systems, so I think I'm on to something. And the expected largest antenna for the retinal, it's smaller than the smallest antenna that ever actually exists, and so the fact that there's no retinal systems with an antenna other than just sort of a extra dye molecule shoved into a gap in the protein, I think I'm onto something. I'm reproducing what actually is seen. And like once again, I can parameterize this however I want. This is sort of the advantage that each system has at different light levels. So once again, the chlorophyll is efficient per unit resource at low resource availability. The retinal is efficient per unit investment at high light availability. And if we want to sort of turn this into a summary, this is, instead of the axis of light, you have efficiency per unit protein versus efficiency per unit light, and these are the two systems. And together, this defines what you call a Pareto Front. So this altogether is the trade-off along which you have to move as something that can compete in the world today. If you start off down here, you're not going to be able to do well. Somebody is going to be better than you. If you're on this line, you can move along this line, but the fundamental structure keeps you from getting better on all axes. In order to get better on one axis, you need to get worse on the other. So this efficiency per protein versus efficiency per resource trade-off is all over microbial ecology, not just this. This is exactly the trade-off that's in respiration and fermentation. This is a nice diagram. In fermentation, you're throwing lots of substrate in the trash, and you get a certain amount of ATP. Respiration, you use less substrate for the same ATP, but you need a lot more protein. There's different forms of glycolysis that have exactly the same trade-off that you find in aerobes versus anaerobes. And when you are maximizing return on a small investment, this maximizes your growth rate in rich resources or your flexibility because you can have more kinds of machinery. And if you're maximizing return on a resource, that maximizes your yield on a rare resource or your ability to be a specialist in some kind of narrow niche. So what does this say about this major innovation? There's extremely precise niche partitioning between these two phototrophs. They neatly divide the phototrophic niche, with one being efficient in terms of investment and one being efficient in terms of resource. And there's other aspects, which I'm not getting into here, but every way you look at them, they're actually really complementary in terms of what makes them up, their ability to horizontally transfer, even the wavelengths of light they use. It's eerie. And I think it's unlikely that these would be dividing the niche so precisely if their properties were random. I don't think they are. And this suggests that there's ecological interactions in their past causing evolutionary priority effects. And that's where something evolves into a new niche, it spreads and fills it, it gets good at that niche, and prevents new upstarts from surviving in that niche because they are going to be very bad compared to it. And I think each mode of phototrophy has been suppressing the evolution of things to like themselves. Now, let's look at that in just a little more detail because the story gets interesting. This is sort of a diagram of the process of evolution based on that trade-off that we saw before. Either one of these is going to start off bad in both respects. And it's going to evolve through some free lunches, getting better in terms of resource and investment until it hits the physical limit. And then it's forced to trade off along this front. The thing is, both of these have a full trade-off front. And so why are there two? You might expect that whoever came first would have just filled this entire front and prevented the evolution of everything strictly inferior to it. And it turns out that there is something that a chlorophototroph can do that a retinalophototroph can't. No retinalophototroph can build biomass using light. It just doesn't pump the membrane up to a high enough voltage to run redox reactions in reverse. So even a very bad chlorophototroph can make a reducing agent that can build biomass. So what this says to me is that I think it's likely that retinalophototrophy had to have come first, because if chlorophototrophy came first, the first awful retinalophototroph would have been worse than chlorophototrophes on all axes. Whereas if the retinalophototroph came first, the first chlorophototroph had one trick it could do, and it was able to evolve and then knock off this part of the trade-off in this energy space. So, in summary, I think this is what has been happening across the history of life on Earth. This is sort of the space of ecological niches. You invent a bad retinalophototroph, it gets better, it fills a bunch of niches. Then you invent a chlorophototroph, it gets better, it fills a bunch of niches, it kicks retinalophototrophy out of some of those niches, but not all, because they're sufficiently different. And both of them have been suppressing the evolution of things to like themselves ever since. And you could imagine very different circumstances. We see something like this on the left with animals and multicellularity. Just more and more multicells keep popping up over and over and over again. They don't seem to interfere with each other or step on each other's toes. You could also imagine a circumstance, though, where something like this happens, but the niche space just wasn't complex enough for there to be space for more than one, and somebody fills the entire niche and completely suppresses everyone for all time. So, conclusions, I think phototrophy is easy to evolve. It originated independently twice from co-opted non-phototrophic systems. You can see some of the things it probably came from in the case of chlorophototrophy. I think this should be very early in Earth's history and be ubiquitous in any other biospheres. I think that this whole dual singularity aspect suggests that major innovations can rapidly become unbeatable incumbents that leave unrefined upstarts unable to compete. And I think retinalophototrophy likely evolved first for reasons that I said before. And I think other evolutionary singularities might be singular due to these priority effects rather than difficulty or contingency. And I think we need to look carefully at the niche structures that can cause this phenomenon sometimes, but not other times. And I think eukaryogenesis, origin of genetic code, origin of life are all prime candidates. I have thoughts on how there might actually be little bits of evidence of other things approaching the complex cell architecture of eukaryogenesis, which I would love to talk to people later. And I have ideas. So, and this has even inspired some lab work. I'm now putting rhodopsins into yeast and studying how it's affecting the evolution of their multicellularity. It turns out there's interesting experimental stuff to be done with this too. So I'd like to thank you all for your attention. I think Will Ratcliffe for letting me go off on these tangents, Ozan for constantly inspiring me in the lab, and Autumn for doing some of these cool experiments with rhodopsin that I've just started recently. So any questions?
Thanks, Tony. That was really exciting, and thank you. Beautiful talk. I have a question about the retinol allotropes coming first. Don't you need chlorophyll first in order to make oxygen, because retinol requires oxygen to be made?
Retinol does need oxygen to be made. Thing is, I don't think that necessarily tells you anything, because you also have chlorophyll getting made with oxygen in a lot of organisms today. And secondly, today, in this oxygen-soaked world, the retinal organisms, they do a lot better in bright light. Is there a place on Earth that has bright light that doesn't have oxygen today? So, yeah.
Yeah, well, you want me to
but, yeah, we'll talk, we talk about that, yeah. Yeah.
Very low oxygen.
Uh-huh. Yeah, and so if the other thing is, though, a pathway that uses oxygen to make this is going to be a lot shorter and simpler than a pathway that doesn't. So if you consider the synthesis machinery for the pigment as part of the machinery, you also get a major advantage if you can cut down on that synthesis machinery. So, but for point 3. I don't know that anyone would recognize a non-oxygen using pathway to make that if they saw it. Like they've characterized the oxygen using pathway. You might just see, oh, this thing has a rhodopsin, but it doesn't have a synthesis pathway. Like are we sure that we have not actually seen that? So between all these things, I'm not sure it says that much.
So Tony, it seems like the really important aspect of determining how many origins of a major transition is its ability to spread and fill an ecological niche.
Uh-huh.
Is there any way that you can sort of suss that out? How quickly can it spread, or did it spread, that would allow you to sort of test this hypothesis?
So, is there a way to look back into the history of a particular major innovation and see how quickly it's spread?
Well, in particular, this one, is there any way to determine from existing, geological evidence or genomic evidence how quickly it did spread in its ecological niche?
Let's see. You might make an argument about stem lengths, about with stem groups and crown groups. There's the crown group, which is everything that's around today and its common ancestor, and then there's the stem, where it was still doing something like today, but then only one thing appears, because none of these innovations are a single step. There's lots and lots of steps, and presumably you're on the stem as you're approaching the trade-off, and then once you're on the trade-off, you start having diversity that remains around today. So I suppose you might be able to make an argument about how big are those stems, how much had to happen along that path, and how quickly did it have to go there? So that's more about the speed with which you approach it. But I think I've thought about major innovations like eukaryogenesis, where you have this enormously long stem with so many things happening along it, and lots of people like to say that it had to take a long time. I'm not so sure. Evolution can happen fast, especially when there's major selective advantages. But yeah, I think that's how I would think about approaching it. Looking at the stems and when the stems had to happen and what had to happen in the stems.
Great, and then one last question.
Thanks so much. I had a question about the evolution of the chlorophyll system, that it seems like it had a lot of parts, and that early on it probably had fewer parts. And did those parts look like the rhodopsin parts, at least in cartoon form?
They don't look like the rhodopsin parts at all. The rhodopsin is this little small transmembrane protein that just literally mechanically is pushing protons around as the pigment changes shape in response to light. Literally, a double bond switches from cis to trans in response to absorbing a photon. The chlorophyll stuff, it's part of an electron transport chain. You are boosting electrons to different redox levels in response to light, and then they're tunneling around. And so,
But do you think the antennae came first, or the motor?
I think. What do you mean?
Well, you had the different parts, the components in cartoon form. You had the antenna, and you had the motor, and then the rhodopsin one looks simpler.
The interesting thing, when you think about their pre-dependencies, for rhodopsin, all that you need is a membrane with a proton gradient that is used to do work. Like it is an advantage in that circumstance. For chlorophyll, you need electron transport chains, basically, and you can also make the argument that's more dependencies for the one to work than the other. Does that make sense?
It's really interesting. Thank you.
Great. Well, thank you.